So apologies, everyone. I mean, I was supposed to have given my research talk, but since I was so slow this morning, and I only covered half of what I really wanted to cover, I've decided to just continue with that. But what I want to go in, so I want to first finish where we were with these reservoir hosts and the characteristics of the reservoir host that So I know it was a bit messy, so I just wanted to quickly summarize what we have done until now. So the first is that we kind of said that, okay, fine, there is this kind of naive host right in the center, right? And there is this reservoir host. There may be some kind of an amplification host. And obviously, the minute that you're talking about a disease, you have to talk about the parasite and you have to talk about the environment, right? So these are the things that we have to be talking about. So we first started with what this parasite is and what are the characteristics of the parasite that actually lead to or increase in risk of spillover, right? So the first is, of course, virulence. Then we talked about persistence, that is how persistent it is in either in the host or in the environment, right? And then we talked about mutation rate and the niche breadth. It was pointed out to me later that why this whole niche breadth came was actually not made clear, which is which is the whole point. So niche breadth is one of the most important aspects, like predicting the spillover, right? So if you have these parasites which are highly host specific, they are unlikely to move to a new host, right? It is only these parasites which are capable of infecting a large variety of hosts that will be able to spread or will be able to spill over to a new host, right? This is that so-called host switch event, right? So what you have here is that you have so I put a phylogenetic tree at the bottom. So you have some kind of a phylogenetic breadth of hosts that the parasites can infect. This head star is one novel host in the population. You can see that none of the specialist hosts are capable of infecting this particular new host, but this generalist host can infect that new host, right? So you can see that this generalist parasite can kind of emerge in this new host, right? So if you want to plot your phylogenetic host breadth with your risk of spillover risk, right? You'll see that there's going to be an increase. So basically, parasites which have a high phylogenetic host breadth, these so-called generalist parasites, are the ones that are more likely to spill over as compared to the specialist parasites, right? Okay. Then we talked a bit about our transmission routes and how that's going to be affecting spillover risk. And basically, when we look at the number of zoonotic parasites, mostly about 25 to 40%, depending upon the thing, are vector-borne, and then it kind of falls off oral, direct, and aerosol. Then we move to the host itself, and we've covered three major aspects. We talked about parasite diversity, right? And saying that, orders which are more diverse tend to have more zoonotic pathogens. But then we also looked at this so-called phylogenetic distance. So basically, reservoir hosts which are more closely related are more likely to harbor pathogens which can switch to humans as compared to those which are very far apart. So, for example, ungulates are less likely to harbor a zoonotic pathogen as compared to a primate. We also looked at this anti-parasite defense. So basically, hosts that invest in tolerance rather than resistance are likely to be more permissive. They're more likely to be good reservoir hosts, which have a high intensity of parasites, a high diversity of parasites. So bats, for example, tend to invest in tolerance rather than resistance, and that's thought to be one reason why bats are such good reservoirs or reservoir hosts. Finally, I wanted to talk about this thing of spatial overlap, right? So when we're talking about disease, right, the host or this reservoir host and this target host or the human or whatever it is have to come in contact, right? So Ashwin used the word sympatry. So they have to be sympatric with each other, right? So you can imagine that a host that is sympatric with humans is more likely to have pathogens that can jump to humans as compared to hosts that have never seen a human before, right? And this is just because like if a host keep on seeing humans, there's more chances, right, that a virus can evolve to jump to this new host, which is keep on seeing. So when we talk about this spatial overlap, I wanted to talk to you about three things. So one is, so what are the animals that we are most likely to overlap with? Rodents, are you sure? Just in general? Very good. So rodents are good. Okay, so these are, so rodents are good, right? So these are these so-called commensal organisms. These are, anthropophilic species. They love humans. So they survive very well in human dominated landscapes, right? And so yeah, so that is definitely there, okay? Domestic animals, right? Domestic animals are probably the most important, right? And then we have wildlife. So again, if we just use the same kind of a metric like the ratio of zoonotic hosts to total diversity, right? There's basically what is the probability, right, that this particular thing will be a zoonotic host. If I just pick a species at random, right, you will find that is this based upon this? One second. Yeah. No, sorry, I don't know where my notes are. I think maybe up there. Thank you. I just want to make sure that my axes are correct. Okay. Yeah, so it is correct, okay. So if I just look at zoonotic host versus the total, so the probability of it being a zoonotic host, you'll find that domestic animals are the most likely to harbor zoonotic pathogens. Rodents, I won't just say rodents, I'll say commensals, right? So commensals, they can be rodents. Many bat species are actually very good at occupying agricultural areas and everything else. So all of these kind of species are going to be somewhere here, and then generally wildlife is pretty low, right? So if you look at this, right, about 25% of emerging zoonotic diseases are actually from domestic animals, right? 25%, right? And then the rest, 75%, are from your commensals in wildlife. So you may say, well, actually, that seems as if commensals in wildlife are much more important, right? But again, I point out, you have to look at the species diversity, right? How many domestic animals are there at the most? There are probably around 40 species of domestic animals. The Food and Agriculture Organization recognizes like 38, right? So 40, okay. Most of these domestic animals are either mammals or birds, right? So if I take all the mammal and bird diversity out there, including all my commensals and everything else, I'll probably get about 18,000, right? So basically, this is what? 0.2%, right? And this is 98.8%. So 0.2% is leading to 25% of emerging infectious diseases. So you can imagine the importance of domestic animals in this whole cycle, right? So, okay, so let us, okay. So now, what I wanted to next kind of talk about is the effects of environment. And the effects of environment, if we just look at the effects of environment, they're pretty straightforward. You know, you can say, okay, fine, temperature, rainfall, right? All of these affect disease. No big deal. So rather than focusing on that, what I wanted to focus on was what I like to call anthropogenic effects. Right? So basically, how do human-mediated changes to the environment directly or indirectly affect the risk of spillovers? Right? So this is what I wanted to concentrate on for the rest of this kind of talk. Okay. So I know that this is probably this thing, so I'll just put a star here, and I'm going to move this star over here, and I'm going to start from here. So anthropogenic so in veterinary college, right, they teach you how to write badly, right? So this is not my fault. Every doctor and veterinarian has bad handwriting because it's drilled into you that your prescription should never be like deciphered by like the normal public, right? So that's why my handwriting is bad. I had beautiful handwriting. Really? Great. I think that's why I'm no longer a vet, and therefore my handwriting is good. See, so ecologists and evolutionary biologists are very good at these just so stories, right? So we have to be very careful, right? When we start looking at things from an evolution perspective, you should read this paper, which is called The Spandrels of San Marco, right? It's a beautiful paper, but if you haven't read it, I would definitely suggest that you do. Okay, on that note, what do you think are the factors, like these anthropogenic effects just to the environment, which are likely to affect, let's just focus on spillover risk, not disease in general, but the risk that we will actually have an increase in a spillover event from some kind of a wildlife to a human population. Okay, so what do you think those will be? Habitat loss, very good, okay? So we can say habitat loss. I'll probably just put fragmentation also right here, right? Because generally, the two kind of go together, and you can't really, like in natural environments, you can't really differentiate between one or the other. Very good, okay. What else? Urbanization, very good, okay. What else? Land use change, okay. But what else? I mean, so urbanization is a type of land use change. Habitat loss is a type of land use change. So I can probably say, put land use change right on top, right? And then I can say urbanization. Somebody said agriculture, very good, right? Agri. Very good, perfect. So we have urbanization, we have agriculture, right? And actually speaking, if you take these two alone, right, they are the primary drivers of habitat loss, right? So we have, these are the major drivers. Okay, what else is a major driver? Globalization, very good, okay. Globalization, I'll put globalization over here. Okay, wonderful. What else? Climate change, great. Global climate change, GCC. Okay, great. Let's just focus on these three for now. That's plenty, right? We don't need to go much further. Okay. Let's just start with climate change. How do you think climate change is going to affect disease spillover risk, right? Okay. What is one of the major effects of climate change? Sorry? Temperature rising. Temperature rising, fair enough, right? So before it was called global climate change, it was called global warming. Do you know why we decided that to change the name? Because the effect is not only on warming, but also a lot of other things. Very good. So it was more of a political thing, right? You can't really convince somebody in temperate USA which is going through the coldest winter, that the globe is warming, right? They say, oh, this is all fake news, bad science, whatever it is, right? So what happened was that what is happening is, on average, the global temperature is increasing, but along with this average increase, there's also increase in variability of temperatures, of rainfall, of everything, right? But increasing temperatures is one very important aspect, right? Okay, so how will increasing temperatures affect spillover risk? Changing seasonal effects. Oh, this is very good, okay. Okay. What else? Sorry? Vector dynamics. Great. Okay. So I'm going to put vector dynamics here somewhere. Right? Because you can also change your zoonotic host dynamics. Okay, so why are these going to be changing? Why are vector dynamics going to change due to a warming? Very good, right? So why are these species changing habitats? What are they tracking?
Resources.
Are they tracking resources mainly?
Also for microclimatic.
Perfect, right? So many of these species are actually tracking the climate because the resources are tracking the climate, right? So what they're doing is, this is what is called this climatic envelope model. It's a very simplistic model. Lots of people are now questioning it and everything else, but for the purpose of this talk, it is a sufficient model, right? So basically, what happens is that you have this kind of range expansion or contraction, right? And this is going to be affecting both our vectors and is going to be affecting the zoonotic host, okay? So in terms of spillover dynamics, right? So of course, one very simple example that I can give you is, I'll just take from our mountain ecosystems, is that, for example, if you have a mountain here, right? And it has a certain temperature, and there's a mosquito which can only survive below this temperature, right? And then there's a host that is here, like a Sholicola albiventris, right? This mosquito is never going to come in contact with that host, so this particular host is never going to see the pathogen that this mosquito is going to carry, right? However, if you have even a 1 degree Celsius increase in your temperature, right? What will happen, right? Remember, the Sholicola doesn't really have anywhere to go. That's it. This is the top of the mountain, right? It either has to go extinct or it kind of just hangs on, right? So now the Sholicola is at the very edge. So now this temperature thing has now moved here, and now the Sholicola is here, but now the mosquito has come here, right? And now you suddenly have a novel vector coming in contact with a naive host, right? And this could lead to spillover. This is exactly what is happening in Hawaii. Farah has been working on this on these avian malaria systems for a very long time. But in Hawaii, when avian malaria was introduced, the birds were completely naive, right? And so when it was introduced, it caused a huge amount of mortality. But what they were seeing now is that as these mosquitoes move up in the mountain, naive birds in high altitudes are suddenly seeing these vectors for the first time, and the mortality is increasing there, right? So this is one way in which climate change can actually affect spillover risk, right? The same thing happens for our host dynamics. As these zoonotic hosts move, they may come in contact with human populations that they never come in contact with before. For example, they're showing in North America, plague is carried by these prairie dogs, right? And as the prairie dogs are either moving up in altitude or up northward in latitude, they may be coming in contact with human populations that have never experienced plague before, right? And this could cause emergence. Of course, there's always been a low prevalence of plague every now and then, but it could cause a kind of a resurgence of plague in the US, right? Okay. But more important, I'll come to seasonal effects in a bit. I just wanted to also say that there are going to be direct effects of temperature itself, right? So there was a paper that came out in, I think, Nature Climate Change, which was predicting that approximately 50%, half of all known human diseases are going to be aggravated by climate change, right? And there are multiple routes that this can happen, but one route is in terms of either your host or vector competence, right? What do we mean by that? How good is that host or vector in either amplifying or maintaining that particular pathogen, right? For example, there was a paper that came out from Kazakhstan in PNAS that showed that a 1 degree increase in temperature will increase the prevalence of plague in Kazakhstan by 50%, right? Why? Because the gerbil, which is the primary reservoir host, becomes a more competent vector when at warmer temperatures, right? The same thing happens for vectors, right? But in the case of vector-borne diseases, there are two factors. One is that as temperatures warm, the vector generation time usually shortens, so you can complete more generations per year, right? So you have an increased population of vectors. The second is that we have a term which is called the extrinsic incubation period. That is the time it takes to go from being infected, so it takes an infected blood meal, to being infectious, right? Lots of data showing that as temperatures warm, the extrinsic incubation period is going to shorten. So vectors are going to become infectious quicker, right? So both of these kind of things are going to make these hosts and vectors more potent, either as vectors or thing, to lead to increase in emergence of these zoonotic diseases, right? Seasonal effects are a very good point, right? And I want to mention only one aspect of it. And that is that in many cases, right, I'm not just talking about seasonal effects, I'm just talking about this increase in variability, right? So, so for example, or increased variability. Okay. Right? So you can imagine, for example, that if you have a situation, right, where you have to have, say, a certain temperature or something of that sort for a population to reach a certain critical size to maintain the disease, right? A seasonal population will go something like this, right? However, when you have increase in variability, right, you could get a population that has increased amplitudes, right? If, for example, there is a certain threshold that the population has to cross to be capable of transmitting the disease to the target population, right? If it was lower amplitude, it is going to rarely hit that high point. However, as the variability increases, every now and then you may get these like sudden booms and sudden busts of these populations, and you could get a situation where you have more and more chances of these kind of, or more and more opportunities for these diseases to emerge, right? Okay. Global climate change, temperature, range effects, seasonal, okay. I think, I don't think I've forgotten anything. Let's move, if I remember anything else, I will come back to that, but let's talk about urbanization. So Abhi just talked about dogs, right? Rabies, very good example. Urban areas, right? There's going to be a increased aggregation of these dogs in, for example, in these trash dumps or whatever it is, right? So that is one way in which, so what is called resource aggregation. You can see that this is a small subset of the various ways in which these kind of environmental effects can lead to increase in spillover, right? But the hope is that we can see that there's a lot of interacting and moving parts, right? So as you have more and more resource aggregation, so for example, trash piles or something like that, you have more and more dogs from different areas coming to the same place, you have more and more likelihood of passing on this kind of rabies, right? Abhi was right that rabies is not density dependent. Rabies is what is called a frequency dependent disease, right? So a single dog in a population could be just as effective, right, as a certain proportion of infected dogs. In the case of SARS, the proportion of infected individuals is the most important. In a frequency dependent disease, it's not. Why? Because of the behavioral characteristics of rabies, right? Because the dogs tend to go out and actually bite other dogs or bite humans. So there's an active component in it, right? Okay. So resource aggregation. The second aspect I wanted to talk about, and which is a very exciting area, is changes in your community structure. So Rory Gibb, yes? Yes, so we will talk about that when we come to globalization, right? It's the easiest way to talk about invasives. Okay. Rory Gibb, who's going to be coming, actually published a paper in 2021 in Nature, right? And it was such a cool paper. And why was that? The paper is a very nice paper. I've put it in that folder that I'm supposed to give to this thing. Again, I'm going to just use this same metric that I've been using. They use a different metric, so but the idea is the same, right? So zoonotic host versus total diversity, right? So the number of zoonotic hosts versus the total number of hosts in a particular area, right? What they found was that, so if you have this as say 1, right, where the zoonotic hosts are exactly equal to, 0.5, right? So this is exactly 50%, right? Then there's equal number of zoonotic and non-zoonotic hosts, right? What they found was when you looked at natural areas, you actually found very low proportion of zoonotic hosts, right? However, when you looked at urban areas, the number of zoonotic hosts is actually more than the number of non-zoonotic hosts. The reason for this, or so they posit, is that the hosts that tend to kind of inhabit human-dominated landscapes are also the ones that tend to transmit zoonotic disease. Rattus norvegicus, mus musculus, right? These are all commensal organisms that tend to transmit a lot of diseases, and they also survive really well in these kind of human-dominated landscapes, right? There's another aspect though, right? And that I want to kind of bring up this thing of resistance and tolerance, right? So if we look at this thing of generalists and specialists, this concept of generalists and specialists, what kind of species do you think is going to be more likely to survive in an urban area? A generalist or a specialist? Generalist, right? So we are all clear about that. Why? Because there's so many different varieties of kind of like source of food and everything else. So a dog is a generalist, a crow is a generalist, right? You rarely see a forest owlet sitting in the middle of Bengaluru, right? Why? Because that's a specialist. Okay. So I don't want to say generally, because we are talking about generalists, but usually, when you talk about generalists, you're talking about species with a R-selected life history. Does everyone know what R and K selected life history is? Sorry?
Number as well as the size of.
Is it, number and size, you're getting there. So the K is definitely related to the size, but what is the R? Okay, so it's the intrinsic reproductive rate, right? So this is based upon this logistic model of growth, right? So the logistic model of growth is a very simple model. It basically says that if you have a population, it's going to grow something like that, right? And the reason that it starts leveling off, why does it start leveling off? Resource, right? So you hit your resourcing, and this is what is called your carrying capacity. Which is given by K, right? And this rate of growth, right, is your R. So species whose populations are predominantly like this boom and bust, right? So basically they're taking advantage of very ephemeral resources. They tend to be R-selected, right? R-selected species have a lot of traits. They have short lifespan, have very large number of offspring, right? So these are the species which in the physiological literature, they call fast pace of life, right? So they have a fast pace of life, and these are organisms which tend to survive in human-dominated landscapes. The K-selected species, so like an elephant, for example, right? Very long-lived, very slow reproduction, probably produces what, one offspring every 2 or 3 years, right? Rarely, I'm looking at Arun because he's the expert here. I'll just make up numbers, but it'll be close. So very slow reproduction, right? These are the so-called K-selected organisms. They're going to be tending to stay at that carrying capacity, right? They're not going to be moving very much further from that carrying capacity. They're primarily going to be limited by that thing. But if their populations fall, they're unlikely to make it up, right? This is why K-selected species are going to be going extinct, while R-selected species tend to survive in human-dominated landscapes, right? Okay. There's a huge body of literature that is coming out now that is showing that K-selected species that R-selected species tend not to invest in resistance, right? Why is that? Because they're putting so much of their effort into reproduction, right? That they cannot afford too much extra resources to be putting into resistance, right? They also cannot afford too much on immune compromise, on immune pathology and everything else, right? So generally what happens is that R-selected species tend to have low resistance. They tend to invest in tolerance mechanisms, right? And there's a very simple reason for that. If you are going to be in an environment which is changing all the time, right? You are better off investing in a generic like defense mechanism like tolerance, right? How does tolerance work? If you get infected by malaria, you eat more, you get more resources, you increase your survival, right? You get infected by filaria, you eat more, you get more resources, you increase your survival, right? It's a very generic method, not very effective, if you have a highly pathogenic organism, but effective for most things. K-selected species are very long-lived, right? They're likely to see the same pathogen for many, many years. So it makes a lot of sense for them to invest once in a few of these things that they're going to be seeing, have very efficient mechanisms to deal with that, so that the rest of their life, they're going to be relatively free of parasites, right? Yes?
I hope I'm not jumping the gun here.
No,
But,
no. Jump the gun.
In that case, as we heard yesterday also that bats are an exception to our selected species. But since you are saying tolerance is more in these our selected species, then in that sense, they should harbor more pathogens. But then bats tend to be in the K-selected spectrum and are also harboring a lot of pathogens.
Bats are bats, right? I mean, they just make no sense because they have really high metabolic rates, right? But they are actually, most bats are K-selected. But there is a reason for that also. Bats will actually not survive. So unless they're eating, they're not going to survive. That's why bats go off into like deep torpor. Every night, they just go into torpor, because if they don't go into torpor, they're not going to make it to the next day, right? So they have these physiological mechanisms which can support both this kind of like a very high metabolic rate as well as a long life. Most species that you find, when you have a high metabolic rate, you usually have a short lifespan, right? Torpor. So they basically go almost like, it's kind of like a hibernation, right? But short, like for a night. But it's like a very deep kind of sleep kind of thing, right? It's not very well understood. So their temperature drops, right? Their metabolic rate completely falls, right? Just over the night. And then when the day comes, they recover, and then they go out and they just start foraging. Yeah.
So are there models which have extended the same RNK selection to pathogens from the perspective, because they don't follow this usual path, but are there any models which say that this same dynamics can be applied to pathogens themselves from a bacteria or a virus or something, protozoan, something like that?
So I know that the models for macroparasites and microparasites are completely different. And the reason for that is that I don't know whether I would ever call any parasite K-selected, though I may be wrong. It's a great question. I actually don't know. I'll look it up and I'll get back to you, right? Yes.
Genome sex correlation. It is a small genome, so
very true.
And if, for example, look into DNA viruses, which are highly adaptive and likewise parasites, it works.
Well, actually, Arun brings up a good point. So I wanted to mention this here. So we said that, as mutation rate increases, your chance of spillover increases, but the mutation rate is also highly correlated to genome size. So the ones that have very low mutation rates tend to have large genomes. The ones that have very small mutations or very large number of mutations have small genomes. And the virions have the smallest genomes, and they also have the highest mutation rate. I think there was a paper that came out that said the highest mutation rate ever found is 1 in 400 or something. So it's like crazy high, the mutation rates of these virions. But it's a very good point. Yeah. Okay. Okay. So we've covered these two things. I just wanted to cover agriculture. And in agriculture, there's obviously something which is called AMR. Does everybody know what AMR is? Exactly, right? This is very common. I'm not going to spend too much time on it, but basically we are feeding these animals these antimicrobials all the time as growth promoters to prevent disease and everything else, right? So this is happening like all the time. There was something that I wanted to mention here, which was very interesting to me at least, and that is that when we look at these domestic animals, right, and we plot the number of zoonotic pathogens against your time since domestication, there's actually almost a linear effect. It's like fascinating, right? So basically your dogs, which were like 15K, right, have the highest number of pathogens. Then I think your pigs and your cattle come here somewhere at 10, and then your horses are here at 5, right? So basically, the longer that we've had these kind of associations, the more pathogens they share. But there's another aspect of domestication, right? So as I was telling you, when we're talking about these wild and commensal organisms, the completely wild organisms in undisturbed areas are going to tend to invest in resistance against generalist parasites that they're not familiar with or which are kind of very stochastic. They're going to tend to become tolerant to the specialist parasites where they have a long co-evolutionary history, right? So the parasite and the host are very closely linked, and so therefore there's more chances that they're going to kind of learn to coexist evolutionarily, right? Okay. However, domestic animals buck every trend. And the reason for that is very simple. Domestic animals have been selected to produce. A cow is being bred to produce milk, right? A chicken is being bred to produce eggs. They are not being bred to resist diseases. This is why we have to pump so many antimicrobials into domestic animals, because we've taken out their ability to fight these infections. One, they have very low genetic diversity. Second is that we've selected for them to put all their energy into production rather than into any kind of defense, whether it be resistance or tolerance, right? So this is the reason why we have to keep on pumping antimicrobials into our domestic animals. Okay. The second aspect is that when you get these really high densities of these animals in these so-called factory farms, right? You get this kind of a melting pot. So many of these RNA viruses can actually evolve through recombination, right? So they don't always have to get new strains through mutation, right? So for example, was it the 2009 H1N1? Was it in 2009? Yeah, I think so. So there was a H1N1 outbreak, which was almost a pandemic. It started in the US. The US will never tell you that, right? They made a big fuss about Corona or COVID starting in China. H1N1 started on a farm in the US. But what they found, right, was that this H1N1 was a recombinant of 3 different viruses, right? I think it's a H3N2, H1N2, and some kind of like a Eurasian swine, avian-like swine virus or something like that. But anyway, so 3 different viruses came together because they were in this high-intensity pig farm where you could have these multiple strains of viruses being maintained, and then you get this kind of a melting pot of things. So, and these kind of melting pots can lead to novel viruses, which can then emerge in the human population. Why? Because these farms have a high human population, right? Because these are people who are taking care of the animals and everything else, coming in close contact with these animals. And so you can have a quick emergence, right? The last point I wanted to make as far as agriculture is concerned is, who was talking about bridge hosts today? Somebody, Abhi was talking about bridge hosts, no? Yeah, okay, very good. Bridge hosts. So, bridge hosts are very important, right? So these domestic animals can act as bridges, right? So almost like these kind of amplification hosts that allow for a virus to spill over from a wild reservoir into a human population. A classic example of this was this Nipah virus outbreak. I think it was in the late 1990s, but I'm not sure, right? So it's a fascinating situation, right? What happened was that in Malaysia, they had these mango farms right next to the pig farms, right? So the bats, because they had these mango farms, the bats used to come to eat the mangoes. The mangoes would be half eaten by the bats, fall into the pig farm. The pigs would then eat those mangoes, and therefore they would have an increase in Nipah, right? And this increase in Nipah virus shedding by the pigs led to an outbreak in humans, right? So you can see that these kind of things, horses are another very important bridge host, right? So many domestic animals can act as very efficient bridge hosts for these novel viruses. Okay. I did remember now what I wanted to mention, and that is climate anomalies. And the reason I'm mentioning this is that there was this very cool paper that came out last year in Nature, Ebby et al. So what they showed, and this is I'm always very fascinated by these kind of papers. So what they showed was, I forget now which virus it was though. Hendra, right, thank you. So what they showed was that El Nino 2 years before led to a food shortage 1 year before, and that led to a Hendra, like increase in Hendra shedding in these bats and increased risk of spillover. Why? Because the food shortage led to bats going, so there were two effects of the food shortage. First was that the bat colonies started fissuring. So they started becoming these smaller and smaller colonies spreading out in space, right? Because you have less food. So you cannot all stay in one place, so you tend to spread out, right? The second was that these bats started moving into agricultural lands, and these are the lands where more humans are, and that increased the risk of spread. But the main thing I wanted to say was that you have this El Nino -2, you have food shortage -1, and you have increase in spillover risk here, right? So 2 years before something that happens could lead to a spillover risk today, right? And these kind of climate anomalies are only going to be increasing with climate change, right? So these are all things that we have to keep in mind. Okay. Habitat loss and fragmentation is probably the biggest group of things that I wanted to cover. And I think what I'm going to do is I'm going to put 2 stars and I'm going to move it over here. Okay.
Yeah, before you start with that, I had a doubt here. So let's just say there's an outbreak of a disease. How do you know in the first place that there was some amplification host? And
you don't. So it took almost a decade to find this amplification host for SARS. MERS was very easy, right? MERS, they found the camels were the amplification host very quickly, but to find the civet, it took 10 years. Ebola, we still don't know either what the reservoir host in the wild is or what the amplification host, or even if there is an amplification host. Despite millions of dollars being spent, I'm just making up a word, millions, but a lot of money has been spent on Ebola research. They suspect bats, they suspect a whole lot of things, but we don't know for sure what species is actually the reservoir host for Ebola. In Africa, Ebola keeps kind of popping up and dying out. But again, Ebola, right, 50% case fatality rate, right? So if you're talking about this virulence drop off, right? So I was saying that, like as time goes on, this is actually very bad. Okay, sorry. This is actually time, and this is virulence, right? Sorry about that, right? So as time goes on, your virulence falls, right? But on the other hand, you can clearly see that low virulence viruses are the ones that tend to cause long pandemics, long epidemics, right? So Ebola, 50% case fatality rate, usually very high mortality, but it usually just dies down, right? MERS, 30% case fatality rate, again, very short-lived, didn't spread very far. SARS, about 20%, I think, was the SARS thing. Again, much shorter, less than a year. It didn't have pandemic spread. It didn't go to, for example, it never went to Antarctica, right? COVID is that perfect storm, right? COVID was almost the perfect virus. Why? Because it had a case fatality rate 2 to 3%. It started at 7, came down to 2 or 3%. Large number of asymptomatic people who could go around spreading the infection, right? But it had enough mortality for us to all get scared, right? So that was the reason that COVID became such a big issue. But sorry, I'm diverging from your point. You'll have to remind me of your question.
Yeah,
again.
no, I was just wondering whether there's a pattern if we can tell that, yeah, this outbreak, there has to be some amplification host. I mean, either from...
Not really. I think that, what you generally tend to do is that you go out into the wild and you just start screening and you find the closest kind of potential ancestor. And then if you find that that ancestor is too far removed to be a viable ancestor, then you suspect that this came into an amplification host, kind of became more close to this final one. But then, since you don't know what that amplification host is, you haven't been able to identify where that final mutation took place before it jumped to humans. Yeah.
Just a clarification before you go ahead. So I think in agriculture also the community structure change would come in, right?
Yes. So they did actually do agriculture also. I think they called it something else, less use or more use or something, but it wasn't significantly different in terms of zoonotic versus non-zoonotic. So only the urban was significantly more. It's Rory Gibbs' paper in 2021. It's there in that folder. You can definitely look at it. Again, remember, these are just broad patterns, right? So don't hold me to these. I'm not putting numbers on it. Just broad kind of patterns. Yeah? Okay. Habitat fragmentation and loss. So how do you think habitat fragmentation and loss is going to be affecting? Everyone tired? I'm just about getting started, man. Spillover risk, right? We're talking mainly about spillover risk, so you're thinking about something that's going to increase the contact of a novel host with a human population, is going to increase the shedding of a pathogen, increase the risk of spillover. So these are the kind of things that you're going to be thinking about. Sorry? Perfect,
animals.
right? So habitat loss is going to be increasing your movement, migration of animals, right? Very simply put, right? What is habitat loss and fragmentation? What is one of the major effects? So there was a paper that came out talking about this ecosystem connectivity, right? So when you're talking about kind of spillovers, right? When you have connectivity between two different ecosystems, you tend to have more connectivity. So if you have animals moving between forest and agriculture, so for example, what the landscape that was being shown in Karnataka, right? Where you have like forests and you have like areca nut plantations, you have like some kind of plantation, all of these kind of things, then you have more chances of these animals moving around between coming in contact either with humans or with other animals, right? So that's a very good point. So, but I just wanted to say that, okay, fine, if you say that this, we can just say, okay, fine, these are all can be kind of grouped under edge effects, right? Does anybody know what these edge effects are? What are these so-called edge effects? What edge are we talking about? Yeah.
So basically, when you have like one habitat being lost because of other habitat taking over, you will have this edge where like species itself can spill over from one habitat to the other. So you might see species from habitat A coming to habitat B and habitat. So it's an interface between two habitats.
Perfect. Right? So this is the interface between different habitats. And so, for example, if I have like a patch of forest, say 1 square kilometer, right? So, right? And this is embedded in an agricultural field. What if I go and split this forest with some kind of a road, right? The road is narrow enough that we can ignore the loss of habitat. But you can see that in this case, right, the area of the square to the edge of the square, right, is about 1 by 4, right? So 1 square kilometer divided by 4 kilometers in terms of perimeter, right? Okay. In this one, assuming that the road is relatively small compared to everything else, you can say that the area is still 1, but suddenly you have, so you have this 1, 2, 3, 4, 5, 6, right? So your area to edge, right, has actually reduced by almost 50%, right? This is one of the basic effects of fragmentation. So as you get more and more fragmented, you have more and more edge versus interior, right? And that leads to a whole lot of things. And one thing that it leads to is this, I don't know why I put this here. There was a reason, and I'm going to move it here, okay. And the reason, and that is that as your fragmentation increases, your area to edge is going to increase or decrease? Sorry? Decrease, right? Yeah. But if on the other hand, I say contact with wildlife, for example, right? You'll notice that that's going to increase. Why? Because humans are going to be using this, humans are going to be using all of this. So there's more chances that the organism, in this case is a human, using this habitat is going to come in contact with the organism using this habitat, just because the juxtaposition of these habitats has increased, right? Okay. Next, any other ways that habitat fragmentation and loss can increase your disease risk? So For example, today we were talking about the increased shedding of viruses, right, due to stress. So these are what all can be brought under these so-called physiological effects. So if, for example, I'm going to be plotting, say, habitat fragmentation or loss against, say, stress, as habitat fragmentation increases, will my stress increase or decrease? Sorry? Increase, right? Yeah. So that's going to increase. And is stress going to increase or decrease my viral shedding? Increase, right? So you can see that if I'm going to put viral shedding on this axis, that is also going to be increasing with your habitat fragmentation and loss, right? So you can see that it's not only that these organisms are going to be more likely to come in contact, but because of the stress that they're going through, they also may be more infectious when they come in contact, right? And both of these together can lead to an increase in spillover risk. Okay. Another aspect, which is probably the most important and probably the most debated aspect, is this effect on diversity, right? So if I just think about diversity of, say, these natural species, and I want to plot something like habitat fragmentation and loss against diversity, how do you think this graph will look? Very good, right? So it's going to look something like this. Okay. So how does reducing diversity affect disease risk? Will it increase? So Ashwin actually showed a slide, and this is from Jason Rohr's lab, right? And Jason Rohr is an amazing scientist. Right now, he went to Notre Dame, and he's been doing some amazing work on this, what is it called, the diversity-disease relationship, right? And what they show is that as any good ecological system, the answer is it depends, right? You could have very different effects. So if you say diversity on the x-axis and say pathogen abundance or prevalence on the y-axis, right? You could have a situation where you have this kind of monotonic increase. So as diversity increases, you have an amplification risk, so you have an increased risk of disease. You could have a thing that looks like this and starts decreasing at very high levels of diversity. Or you could have a situation which looks like this, right? Where as diversity increases, you actually have a reduction in your disease. This is dilution. This is amplification. I don't want to go that far. But the most important thing, at least in my mind, lots of people knew that you could have both amplification and dilution. What they showed was that whether you tended, so they did a meta-analysis. Does everyone know what a meta-analysis is? Right? It's an analysis of analyses, right? So they did a meta-analysis, and what they found was that when the study was conducted at scales of 1 to about 1 into 1 million square kilometers, you tend to have dilution. When the study was conducted at scales greater than you tend to have amplification. So most of the time, when we're talking about local processes, diversity protects against disease. When we're talking about very large, so 1 million square kilometers, so they say in the paper, is France and Spain combined. So this is like a huge study going across like one whole country or multiple countries, right? Why is that? It's actually a very simple reason. The reason is that when you go to those kind of scales, you're probably going to be sampling multiple ecosystems, right? And so the rules change, and there is some this thing. If you have time at the end, remind me to come back to why at very large scales you could have a reversal in the overall patterns. Okay, but in general, what you find is that at very small scales you do get dilution. Diversity does protect against disease. At very large scales, you get amplification. But for the most part, those are not the scales of most ecological relevance. Yes, Ansil.
Guha,
Yeah.
as ecologists, can't we say this again? It depends. For example, the biology of the pathogen also often it violate these scale
For sure.
generalities.
100%.
Yeah.
So these are meta-analyses which just tell you the general pattern, right? So on average, if you had to guess, then you would guess that if I was doing a study at less than 1 million square kilometers, if, for example, I was going out and doing a study in, say, in the Western Ghats, and I had to make it a strong study, I have to make a prediction, then I'm going to predict that I'm going to have dilution because the scale of my study is probably going to be like 100 square kilometers at the most, right? So, yeah.
Just to clarify that, so my thought process was basically whether a pathogen is generalist or a specialist. Even in local scale, correct me, Guha, if I'm wrong. Yeah. Should I hold the thought?
Please hold the thought, because I'm hoping to actually have time to go to my study system where we are actually showing exactly what you're saying, right? Okay. So, but the most important and probably the most exciting aspect is kind of depressing but exciting, as most of disease ecology is what is called biotic homogenization. So we said very clearly that as you have more disturbance, habitat fragmentation, loss, you're going to have a reduction in your diversity. But are all species equally badly affected? No. So which are the species which are going to be the most affected, and which are the species which may actually be doing better? Very good, right? See? So if I'm going to be plotting the specialists, you'll get a thing that goes like this. So basically, you have a very steep reduction in specialists. But on the other hand, if I plot the generalists, you may actually have something that goes like this. So the specialist plus the generalist makes the total, right? So what you will get is that about at this point, you'll get a flip, right? In undisturbed areas, specialists dominate and generalists are very rare, right? As you get more and more disturbed, you have a generalist dominating and the specialist becomes rarer and rarer, right? And this is called biotic homogenization, and this has been shown for multiple taxa, right from plants to insects to fish to birds. And what I wanted to say is that our study showed that the same process is happening in parasites, right? And I will come to that if I'm not forgotten anything. I think we're good. Okay. Okay, so I'd wanted to show you my cool slides and everything else, but that's not going to happen. And thanks to Ashwin, you've already seen many of my slides, so I'm never going to let him forget that. And I'll remind him at his PhD committee meeting also. But But yeah. Okay. So what I rather wanted to do, so I am going to upload or send the slides to Ansil, and he can make it available to you. You guys can look at it. What I'm going to do today is give you a broad kind of a thing. I think I have like 20 minutes or so, yeah. And I should be able to finish in that time. Okay. So as I told you, right, when we are talking about free living species, we know that if you have disturbance, you tend to have a community dominated by generalists rather than specialists, right? And this has multiple effects, right from ecosystem stability. Specialists tend to stabilize ecosystems. Generalists are not very good at stabilizing ecosystems. And why is that? Think about a table, right? If I have a table with one big leg, right? So if I have a table that looks like this versus having a table that looks like this, which is going to be more stable? The second one, right? Generalists have a broad niche breadth, right? One generalist can cover the entire breadth of this environment, which is represented by the table. Specialists have very narrow niche breadths, right? You can pack, it's called niche packing, right? So think about this in terms of a bucket, right? Where you're putting in balls, right? If your balls are ping pong balls, you can put in a lot of ping pong balls into that bucket. If your balls are basketballs, you can only put in one or two, right? So again, we're talking about a three-dimensional niche, not this n-dimensional hypervolume, because unlike many mathematicians, my limitation, I'm limited to like 3 dimensions. I cannot think of anything like a 4th dimension. They say time is the 4th dimension. I have no idea. But anyway, so, but you can imagine that if you, so when you have a lot of specialists, you generally tend to get high diversity, right? You get high complementarity, right? So each one is complementing the other's function. So if you lose 1 or 2 specialists, your ecosystem functions will still go on. If you only have one generalist and you lose that, your entire table is going to collapse, right? So there are multiple reasons that this biotic homogenization is very important, right? But from a parasite point of view, it is even more important. Why? So what are the parasites that are more likely to emerge as emerging infectious diseases? Is it going to be the generalist parasite or the specialist? Generalist parasites, right? So if this pattern holds in the case of parasites, right, as you get more and more habitat fragmentation, if you get more and more generalist parasites dominating that ecosystem, you're going to get an increased risk of disease emergence in a novel host, right? On the other hand, it has been shown that parasites are actually a very good indicator of ecosystem health. A paper came out, I think, by McCallum back in some time. I only remember the title of the paper, and the title says, "Are ecosystems that are rich in parasites more healthy?" Right? And they show very convincingly that as parasite diversity increases, ecosystem health in terms of ecosystem functionality actually increases. Why is that? We tend to think about parasites as a very negative thing, right? But we don't think of tigers as negative, right? We don't think of herbivores as negative. Parasites are just like tigers and herbivores. They play an important role in ecosystem function, right? So that is one thing that we've been forgetting. And when you are talking about specialist parasites, these are parasites that are only going to be infecting a few hosts that they've co-evolved with. They are not the parasites that are jumping and causing these emerging infectious diseases in humans. But what you will end up having, if this pattern holds, is that your parasite diversity will in general reduce with habitat fragmentation and loss, but you're primarily going to be losing your specialist parasites, which do not cause emerging infectious diseases. You may be actually increasing your generalist parasites that do cause emerging infectious diseases. And this is what we've shown in a paper that we published in 2019. So a note of caution, and I hope my old PhD student is listening to this. I told Pooja, hey, this is a really cool analysis and you have to do it. She said, no, this, that, everything else and everything. And then she left. And then I was left holding the baby and I wrote the paper. So I'm the first author on the paper. This is the only paper in avian malaria that I'm the first author on. But I think that if you ask me, that was probably the most important paper from her PhD, which she never published. So always listen to your advisor. No. Never listen to your advisor. Okay, so, oops, I have like 10 minutes, so I have to rush. Okay, what we showed first, so in the paper that Pooja published first, so Pooja published the first paper in 2019, right? And in that paper, what she showed, so we're talking about avian malaria, right? Avian malaria is a vector-borne disease. It has two, we're talking about avian hemosporidians, right? Avian malaria is caused by Plasmodium. Avian hemosporidians are the broader term, right? So we're talking about avian hemosporidians. These are vector-borne diseases. You have two major genera that we focus on. We have Hemoproteus and we have Plasmodium. There's also leukocytozoon, but we have so few leukocytozoon in the Western Ghats that we didn't have any ability to analyze it. So we focus on these two, right? And they have very different biology. So for example, Hemoproteus is transmitted by these biting midges. Plasmodium is transmitted by the mosquito, mainly Culex kinkifaciatus. So there are some differences in the biology. But to cut a long story short, what we showed was that, I'm just going to use blue and red because that's what we use in our paper. So red for Plasmodium and blue for Hemoproteus, right? So if we have bird 1, bird 2, bird 3, bird 4, right? Actually, I don't want to do it this way because that is actually not entirely true. Actually, none of it is entirely true. I'm just making up things as I go. But so what we found was that if we look at the Hemoproteus lineages, they tend to focus on one or at most one or two closely related bird genus, taxa, right? However, when you look at Plasmodium, it has a more generalist kind of distribution, right? So they tend to affect multiple species, sometimes not even related species. They tend to be more generic. Okay. Now, there were so this was just based upon this host association test. So basically, you are asking, is the phylogenetic breadth of this parasite greater than expected by random chance or not? And what we found was that in the case of Hemoproteus, almost all the parasite lineages were less broad than expected by chance. In the case of Plasmodium, there were two lineages which were significantly greater than expected by chance. The rest were not significantly different. The reason that most of the Plasmodium was not significant is actually related to this generalist and specialist. Specialists tend to have high prevalence, right? Generalists tend to have low prevalence. So Plasmodium prevalence is about 10%. Hemoproteus prevalence is about 60%, 70%. So we have a lot more sample size. We collected 1,000 and odd birds, but with 10% infection, you only get 100 positives, right? But with 70% infection, you get a lot more positives. So you have a lot more power when you're talking about Hemoproteus as compared to Plasmodium. Okay. But there were lots of other factors that we also saw. One was that, of course, the prevalence, right? The second is I'm just going to cover these three, but I'll talk to you about our results on alpha and beta diversity later. But we are just working on the paper now, so it's not yet published, but we are getting some really cool differences between hemoprotease and plasmodium alpha and beta diversity. But one, we found that hemoprotease had higher prevalence than plasmodium. Second, we found that when we are talking about co-phylogenetics, so basically if you regress the host phylogenetic distance against your parasite phylogenetic distance, in the case of hemoprotease, there's this positive thing. So basically, so as your parasite phylogenetic distance increases, the host phylogenetic distance increases. In the case of plasmodium, there's no phylogenetic effect, right? The third is, and this is the really cool part, when we took the global phylogeny of these hemosporidian parasites, what we found was we took this measure which is called the nearest neighbor. So we calculated the nearest neighbor distance for all our lineages, right? And what we found, and then we randomized these across the whole global phylogeny. And what we found was that the hemoproteus lineages tended to cluster together more than expected by chance, while the plasmodium lineages were just completely random, right? So basically, if you take a global phylogeny and you just put the plasmodium there, that's how they land, while hemoproteus were clustered all together, right? So all of these kind of convinced us that yes, we do indeed have these very different kind of parasites where plasmodium is a generalist and hemoproteus is a specialist. Okay, so then we did what is called a structural equation model. And structural equation models, we did a random forest model also, but random forests are these black box models. They're very good for prediction, but not very good for explanation. But a structural equation model is very cool because it's this hierarchical model where you can kind of model these kind of correlated effects, right? So what we did was that we had two measures of disturbance. One was human population. The second was distance to protected area, right? So as human population increases, your disturbance increases. As your distance to protected area increases, your disturbance increases. We also had our bird diversity. Then we calculated plasmodium diversity, and we classed the same hemoproteus diversity. We also calculated hemoproteus prevalence and plasmodium prevalence, right? And what we found was very interesting. What we found was that distance to protected area has a negative effect on bird, which makes sense. So further you are from a protected area, the lower your bird diversity, right? Bird diversity has a positive effect on your hemoproteus diversity, and hemoproteus diversity has a positive effect on hemoproteus infection risk, right? Interestingly, your bird diversity did not have any direct effect on hemoproteus prevalence. So bird diversity is only affecting hemoproteus prevalence through hemoproteus diversity, right? In the case of plasmodium, it's a very different scenario. Here, bird diversity has no effect, right? But bird diversity actually has a negative effect on plasmodium prevalence. So as bird diversity increases, your plasmodium prevalence is reducing. There's a strong dilution effect, right? While in the case of hemoproteus, there's actually a strong amplification effect, though it is indirect. So bird diversity increases, hemoproteus diversity increases, hemoproteus prevalence increases. There was also a direct effect of human population on your plasmodium and on your hemoproteus prevalence, but they were in opposite directions, right? So in the case of hemoproteus, as human population increased, you had a negative effect. And in the case of plasmodium, you have a positive effect, right? So basically what you're seeing, right, is exactly what we had predicted based upon free living species. And that is that as you have more disturbance, your generalist parasites increase, your specialist parasites reduce, right? And this has broad implications in terms of emerging infectious diseases, right? So basically, I would say that here we were talking about all of these anthropogenic effects on the reservoir host, but similarly, we can probably also put these kind of anthropogenic effects directly on the parasite, right? And I think the take-home message that I wanted to tell you was that parasites are a very good indicator of ecosystem health. Why? Because specialist parasites tend to be the most diverse, right? 40% of Earth's diversity is parasites. Can you believe that? So we are all talking about biodiversity conservation and everything else, and we forget about 40%. And many of you must be skeptical, right? 40%, that can't be right. How come I've never heard of this? Think about it this way, right? There's actually a paper that came out where Hudson et al. Estimated this, but I think about it very simply. You take any species on the planet, it'll probably have at least one parasite which is a specialist, that is only there on that species, right? Okay. So there itself you have, so if you have 10 species, you have 10 species of parasites which are specialists only on those 10 species. Okay, fair. Some of these parasites have parasites. You have hyperparasites, right? So you can imagine that the 40% is probably a huge underestimation of the actual total parasite diversity, right? But the main thing is that most parasites are highly specialized, right? So the large proportion of this parasite diversity are specialist parasites which are not going to be like really important for emerging infectious diseases, right? So parasites are a very good indicator of ecosystem health. They are like all symbiotes, right? Because their biology is so closely tied with the host, they are very sensitive to changes, right? You take away the specialist host, the parasite also has to go extinct. It doesn't have a choice, right? So they are very good indicators of ecosystem health, but generalist parasites are very important from a public health perspective, right? And basically what our data is showing is that when we go out and modify these ecosystems, we tend to reduce the parasites that are not going to cause harm, and we're going to increase the parasites that are more likely to cause harm. So with that, I will be happy to take questions. Yeah, Ansil.
Or something, but previously you said Plasmodium is generalist. It's infecting B1, B2, and all the birds.
Oh, sorry. Yeah, I think I got the colors mixed up.
Yeah, so then I would expect a positive relationship with the bird diversity and Plasmodium prevalence below. No, am I wrong?
So as bird diversity increases, your Plasmodium diversity actually reduces. So there's a dilution. Because as disturbance increases, bird diversity reduces. So in disturbed habitats, you have low bird diversity and high Plasmodium prevalence. In undisturbed habitats, you have low Plasmodium prevalence and high bird diversity. It's the exact opposite in the case of Hemoproteus, where in undisturbed habitats, you have high bird diversity, high Hemoproteus prevalence. In disturbed habitats, you have low bird diversity and low Hemoproteus prevalence. And the Hemoproteus is the specialist. Sorry, I completely turned the colors around. I was hoping that the colors would actually help. I think they actually confused in the end. A specialist. Prevalence. Yeah. So bird diversity doesn't affect Plasmodium diversity. Plasmodium diversity actually does have a positive effect. So as Plasmodium diversity increases, your prevalence increases. So that is common. So parasite diversity increases prevalence both in the case of Plasmodium and Hemoproteus. The only major difference is Plasmodium diversity is not linked to bird diversity, while Hemoproteus diversity is closely linked, right? Because these are specialist parasites. The more diverse your host, the more diverse the parasite. These are co-speciated, so there's no way that this is something that we would expect, right? But again, as Hemoproteus diversity increases, you have an increase in your Hemoproteus prevalence. So on the whole, what we're finding is that as you have more disturbance, Hemoproteus infection rates fall. As you have more disturbance, Plasmodium infection rates rise. Yeah. Yes.
I don't know whether it's just a thought that occurred to me, and I wanted to ask you what you thought about it as well. I mean, based on the effects, I mean, the results of your study, we see that anthropogenic effects directly. There is a direct relation between anthropogenic effects and parasite prevalence. So in that case, going forward, not in the present time, going forward, can we have, we talk about HF, habitat fragmentation and habitat loss. In the future, with more green spaces, the call for green spaces coming in, can we have urban fragmentation and habitat gain leading to more spillovers? Would that be possible?
Okay, so we recently got a paper accepted in Nature, Ecology and Evolution, and this is actually talking about the so-called landscape consolidation in China. And we are showing that basically, as you have more and more landscape consolidation, you actually have increased risk of hantavirus emergence. But this, we're mainly talking about agricultural land consolidation. We're not talking about a real increase in natural land. So basically what China did was that they had these very fragmented kind of small farm holdings. What they wanted to do was to consolidate these into larger farm holdings. And they've been doing that for at least like 25, 30 years now. But what has happened because of that is that there's been a sharp decline in your rodent diversity, right? And the only rodent that is surviving is the major reservoir of Hanta. But I did want to mention that I don't know. It is quite possible that when you have these kind of green spaces in the middle of cities, and you're going to have like these wild animals that are going to be colonizing them, you may actually have an increase in contact between humans and these wildlife, and therefore you may actually increase your risk of disease. We can't rule that out.
But I think it also depends on what the community in that urban space is, right? So basically the problem with a lot of urban systems is they're like completely de novo kind of, they have no clue. I mean, even this idea of, habitat loss, distance from forests, it's kind of still binary and traditional in some sense, right? It's still kind of a gradation from the forest. The further away you are, the diversity decreases. It's still simplistic.
What we also used.
Yeah, no, I'm not saying it's, I mean, it is still based in that idea.
Yeah.
But an urban space is like a new space, completely new community, like new things. And so I think it's a completely different zone. And I think all these decisions of, increased green in urban space, et cetera, what they mean, they may mean something good for carbon. They may mean something good for temperature cooling, reducing heat islands, reducing like water dumping. But what do they mean for, say, disease? I don't think we have a clue. I don't know, maybe I'm wrong.
Very true, very true. So for example, when you look at your, just your mosquitoes, right? If you look at Aedes aegypti versus Aedes albopictus, Aedes aegypti survives really well in completely built up environments. Aedes albopictus survives in slightly greener, so having these kind of more urban green spaces, you may find a shift from Aedes aegypti to Aedes albopictus. Aedes albopictus generally is thought to be a more efficient vector of Zika. Farah, am I right? Yeah, but yeah, but you could have a difference in the kinds of pathogens that are going to be spread, right? And this is the case for many, many things. So for example, we were talking about range expansion due to climate change, and we only are looking at, increases in disease, but actually that's not the case, right? They're actually predicting that due to climate change, malaria in sub-Saharan Africa is going to go down because there's going to be less and less habitat suitable for anopheles, and dengue and chikungunya are going to rise because the habitats are going to become more suitable for Aedes and Aedes albopictus and Aedes aegypti. So these are very complex kind of things, but it's a very good question. The answer is, I know that it took me a long time to get to this. I don't know.
Excuse me. In terms of genetic fitness, host genetic fitness, how have you?
Brilliant point. As you can see, there's a huge body of literature which I've completely ignored, and that is this issue of host genetic fitness. So as you get habitat fragmentation and loss, you generally tend to get these kind of isolated populations. They tend to get inbred more. Inbred populations tend to be more susceptible to parasites and to pathogens, and therefore there could be a real increase in things. This has been shown very clearly in the case of things like the cheetah, in the case of the Florida panther. So when you have something which it's called inbreeding depression, so basically you're breeding mainly with your relatives, then what happens is that you have lower and lower standing genetic variation, and so you're less and less capable of fighting off your parasitic diseases, but you also have more genetic abnormalities, right? Because you have these deleterious recessive mutants that kind of increase in their frequency. But thank you, that's a very good point. So has anybody heard of this term called extinction vortex? So basically, if you have, what they've shown is that if you have habitat fragmentation and loss, these populations become smaller and more fragmented. They'll become more and more inbred. They're going to be more and more susceptible to diseases, to stochasticity and everything else that further reduces the population, increases inbreeding more. And so basically you get this kind of a negative feedback loop, and that just drives the population straight into extinction.
My question is like, in that other case, where the bird diversity is impacting the diversity of this pathogen,
yeah,
then that pathogen is impacting on prevalence.
Yes.
And you are telling this bird diversity is not directly impacting on this third one.
Yes. So, there's no significant direct effect of bird diversity on.
How we can test this thing? Because A equal to B, B equal to C.
Yeah.
Then how?
So this is what the advantage of using the structural equation model is. So what the structural equation model does is that it builds this kind of hierarchical set of regression models, right? And so if you have a strong effect of bird diversity on this and then this, once this pathway is taken care of, basically it takes that effect out, and then it asks whether there's any variance left to be explained with this connection. So basically what it does is that it builds this most parsimonious set of connections, right? They say it's causal modeling, but it actually is not, right? It gives you some idea of causation better than your normal models, but it is very, very dependent upon the hierarchy, right? So if I, for example, switch the hierarchy, I could say that instead of A causing B, I could say B causes A, right? In this particular case, it's very simple, right? Because I cannot ever imagine a situation where hemoproteus diversity, right, or hemoproteus prevalence is going to be affecting the human population, right? Hemoproteus doesn't affect humans at all, so there's no way. So in some situations, the direction of these effects is relatively clear. In those kind of situations, using these structural equation models is good. But the point is that you can basically have these multiple effects, and you can parse out which is the most important, what is the direct effect and what is the indirect effect. I'll be happy to talk to you about SEMs at any time. Yeah. Wonderful. Thank you so much.